Good afternoon, guys. I'm glad to be here today. Today we will be talking on today's section. We'll be talking on how to do protein-protein molecular docking or protein-protein molecular dynamics simulation. And in this video today, we are going to look at using different a particular software, a particular server. No need for download of softwares. No need to install any or download any software. We just make use of freely available server on the internet from Harvard for your molecular protein-protein docking. This is different from protein-ligand docking. This is protein-protein docking. Probably in your study, you are looking at interactions between a protein and another protein. For example, maybe you are looking at interaction between spike protein from SARS-CoV-2 and ACE2 enzyme from host. So you can use this software to do your molecular docking of protein-protein interaction. As you can know, the popular chimera can't do protein-protein interactions. You can only that can only do protein-ligand interaction. So in this video, I'm going to show you how to do protein-protein docking to study protein-protein interaction without downloading or installing any software. All you just need to do is to go to your browser, and once you go to your browser, just type plus pro docking. Actually, there are many soft server. We have the ArchDock, we have the PatchDock, and many of them. But today, I'm going to show you how you can use this Clos Pro docking. Just type Clos Pro docking on your browser, and you can see this is Clos Pro 2.0 protein docking from Boston University. So click on it. So once you click on it, it gives you to the page where you can register. Yes, you can register. So once you register, let me show you. I already did my registration. Then once you register, you can log in. The registration is simple and faster. So you can log in, and once you log in, you will have this page open up for you. So this page, we have dimer classification, view, results, references, download, paper for publication. If you use this for your work, you can cite any of this publication, help, and in case you need help for anything, then for contact, in case you want to contact them as much as you may need. Now, so to do this, your two proteins you want to dock must be in PDB format. Your two proteins must be in PDB format. And once it is in PDB format, so you can download your protein. It could be a protein you download from Protein Data Bank, from any other data bank, or maybe a new sequence from your lab. So once you do that, you can give it a name. So let's call this one example. Then to make it faster, we have the server as CPU and GPU option. So I'm going to make use of the GPU option we know GPU is faster than CPU. And after that, so you can probably, if you want to dock directly from database, from PDB, Protein Data Bank, you can put the PDB code of your protein in this box. So this is the receptor and this is the ligand. You will be asking yourself which of the proteins is going to be ligand, which one is going to be receptor. The protein with the highest number of amino acid sequence will be the receptor, advisably, and the protein with the lower amino acid sequence will be the ligand. So once you do that, you can just put the PDB code of the protein if you know it, and PDB code of the ligand if you also know it. But if you don't know it, you can simply go to, you can upload that document and use that PDB file from your local laptop, local disk on your laptop. So in this case, let me use this as example. For example, then I can also go to my ligand, fuse ligand. Then let me use a protein. Let me just use a protein here as example. So once you choose that, not necessarily you put the chain number. No, you don't need to put the chain. Just leave it that. So you can, once you have identified, you've selected your receptor, you selected your ligand, then the next thing is there are some other options like advanced options like attraction and repulsion. So what is attraction and repulsion means, if maybe on your protein, maybe you want a particular segment of the protein to be attracted to just another segment of the receptor of the ligand. Maybe there is amino acid 1 to 50. You want it to bind with amino acid 100 to 150 on your ligand. You can indicate. So whatever binding you are doing will occur between the amino acid you selected on the receptor and the amino acid selected on the ligand. And this other segment is the repulsion is showing you, okay, maybe you don't want a particular segment to interact with each other. You don't want a particular segment to dock with each other. You can indicate that particular segment on this. So another thing, we also have the structure modification. Maybe you want to modify from the receptor or from the ligand. And there are some other options for antibody mode, other mode, as many as you can think of. So once we've done that and you've selected, don't forget, let me do this again. You can give a job name, select a GPU option to make it faster. We have GPU and CPU option. Then you can now indicate which ligand is your receptor, which protein is a receptor, which protein is the ligand. And like I said, adversibly, the protein with the high amino acid sequence should be larger amino acid sequence should be the receptor, while the protein with the lower amino acid sequence will be the ligand. And otherwise, you can also choose this from your local desktop, local disk, or you can use the PDB code for this. So once you have done that, the next thing to do is to click on dock. So once you click on dock, can you see? So this job is processing on the server. It takes some minutes, close to attempts hour before this can be done. After this is done, then you can come here and find your results here. So let me click on this finished results, and you can see, so it's going to give you the display mode. You can choose, maybe you want the best, based on the best quality. Maybe you want the best 10, the best 15, the best 20, the best 24. So in this, we are choosing the best 10. And as you can see, this is 1, 2, 3, 4, 5, 6 now. So we have up to 10 of it now. So that is so on this, you can download any of these ones that you want. But let's go and look at let's view the model score. Can you see for the cluster 1 is 0? This is the weighted score. And as you know, for this molecular docking, the more negative the value, the better the docking and conformation that we have. So on this cluster 0 means as minus 1144.2 at the center and the lowest energy. So you can see, so on this, it depends. You cannot decide which conformation do you want to choose. You understand? And this is the calculation this is the formula for calculating this weighted score. And you can also look at the electrostatic of this. What is the electrostatic value of each of them? What is the hydrophobic favor value? Can you see? What is the van der Waals electrostatic value? So this can give you information about the binding energy that occurs between your protein receptor and your protein ligand. So once you do that, you can download that, like I've downloaded this, and you can use any of your visual software, maybe Chimera or anyone that you use to view this. So let me see in this case. So let's see this. So can you see, this is the receptor. Can you see the receptor here? And this is the ligand. Let me show you that so that you can see. This is the ligand protein. and this is the receptor. So once you're able to do this, you are done. So from there, you can continue looking at other factors that is, as important to you in your molecular docking. And don't forget, once you're able to do this, you can just go to, this so that you can cite this paper. You can cite any of these papers, as a reference for using this server. So the CLOS Pro is very good and reliable when it comes to protein docking. It's easier and faster. It's easier and faster. So that's the information for today. Thank you very much. If you find this video interesting, please kindly subscribe to our channel for more information on bioinformatics and other computer-aided drug design and other computational techniques and tools. And if there is any topic of interest that you think you want us to cover, please feel free to send it to us. I will definitely cover that to you. And if you have any question, please comment, like, and subscribe to the channel. Thank you very much.